So Dave Dawson, you may come up with your e-guitar with the sound we have right now. So thank you very much for coming. It's a great pleasure and an honor to introduce Dave Evans. Dave, it's almost don't dare to do this really, because when you look at his CV, not only it's impressive, but there are these pieces like being the president of MDI for almost 10 years. That's very impressive, and steering the lab through difficult. I think it must have seemed like 15. Okay. So Dave went to school in Indiana, and even at that time in college, he was interested in history. And I think you're coming back to where you started originally, and then he studied biology at Stanford, and then he went to Florida, and he went through the ranks at Florida as a biology assistant, associate professor, and then I don't know how this happened. You changed from biology to zoology, and you spent time in other marine biology labs, so I'm afraid to say. So when you started in 1965, you first went to the other lab down the coast. What I can't find out really from your CV, you have been associated with MDI for a very long time, but it doesn't tell us when you first came here. So this wonderful experience setting foot on MDI, I think you'll come back to this during your lecture. After a successful career in Florida and even more successful career here at MDI, Dave is now the historian of the lab. He has written the book. Who knows about, I mean, I don't ask you if you have read it, you know, that's another question, but who actually has seen the book? What are you doing with the second copy? Sorry about that. I shouldn't ask questions like this. But I also own several copies of that. And to be honest, it's fascinating. I'm at first as a work of scholarship. I mean, it's wonderful. It really covers the whole story of the lab. And I have to admit, I read it twice, Dave. I have to admit, the first time I read it, I just left all the administration stuff out. You know, I mean, I just went back where the pictures are of people who have worked here and about the research and all that. And this was the fascinating stuff. However, after I became president, I'm very grateful for you, because I went back, and this helped me a lot to understand what happened, because you are meticulous in describing what happened during difficult years here at the lab, what happened in better years, and covering the whole history of the lab makes you understand how wonderful MDI is. So we are very grateful for your scholarship. We're also grateful for now that you're starting with individual researchers. Unfortunately, I don't think I will become old enough to see you writing about me here at MDI, but there is always hope. So I won't talk for 20 minutes. I think it's all yours, and we are looking forward to Milestones at the MDI Biological.
Crap. There. Unfortunately, these reflections are nasty. That's a little better. Come on, hold up.
And worked in the early years at the Harpswell Laboratory, or at the Marine Biological Laboratory at Woods Hole. Harpswell is just east of Portland. I think I'm getting it, or maybe not. There you go. Harpswell is on a neck, Harpswell Neck it's called, actually, extending into Casco Bay just east of Portland. Here's a better picture of it on the right-hand side. The laboratory, actually, the original lab, which I'll show you in a minute, was up here, and then they moved after a year down to this site, Graveyard Point, which is kind of an interesting title. This is going to add to my stress immeasurably. Okay, this is a flyer for the first summer, 1898. We'll get back to who Kingsley is in a few minutes. Kingsley was the first director from Tufts. He was sort of the father of that laboratory. And this was the flyer. And the first summer they spent in this house, which was called the laboratory, which was just a rented cottage. And the lab was in the first floor, and the students lived on the second floor above all the smelly fish that were brought in every day. The major problem with this was it was about 2 miles or maybe a mile from the water. So to bring up seawater and to bring up specimens was a real pain. So they only taught one summer, 1898, and then 2 or 3 summers later, they finally moved. I should show, here's the cottage in 2011, it still exists. And here's another view of the point. And the laboratory in 1902 had a separate building, which might look familiar to you. And they built a cottage for Kingsley. And these two existed on the lab point. The trouble with the lab point was that this was a graveyard here and a marshy area here. But the good news was there were lots of cottages and hotels here and down here for the students and faculty to stay, although some of them stayed in tents. But there was no place to expand. I don't think that was really too important to them, but eventually it probably became kind of important. And again, oh, next, Roy. Oh, God, I love it. This is fantastic. 1902, that's what was there. This is what's there now. My wife and I visited there in 2011. A young family owns the cottage, and that's the Kingsley Cottage behind it, so it still exists. Next, please. These are the founding fathers. J. Sterling Kingsley, what a wonderful name. John Sterling Kingsley, Herbert Neal, and Oliver Dahlgren. I'll talk briefly about them each in turn. Next. Kingsley got his PhD from Princeton in 1885. He worked on the embryology of horseshoe crabs, so he was a real marine biologist. Typical embryology was a big thing in those days. It still is now, but it was a really big thing in marine science. That's what he worked on. Next slide. Late 1880s, he was instructor at the MBL at Woods Hole. That's the MBL connection. Next slide. 1892, he was chair of biology at Tufts. And then within 6 years, he had started the laboratory in Harpswell. Next slide. So in 1898, he established the lab. Next slide. 1919 was his last summer at the Tufts Marine Laboratory. He never made it. Next slide. He never worked at the MDIBL. He never came up here. He was, next slide. He was to come up in 1930, but he died in 1929. It's kind of romantic. He was on a world cruise with his daughter, and he died, as I said, 3 days out of Yokohama and was buried at sea. So kind of a good ending for a marine biologist, I think. But he was the only one that did not work at the MDIBL. Next. Herbert Neal did. He was our second director. Next slide, please. He got his B as a Maynard, got his BA at Bates. Next slide. Got his PhD at Harvard in 1896, nervous system of dogfish shark. So there was his dogfish shark connection, which continued work here. Obviously, we'll get into that in a few minutes. Next slide. He was at the Tufts Marine Lab from 1904 to 1916. He was the director from 07 to 16. Between about 16 and 1960 and 20, it's not clear who was director. He was gone. And Kingsley was gone, and it probably was Dahlgren, but no, I'm not really too sure what happened. Next slide. He was at the MDIBL from 22 to 40, and he was the second director, late 20s, early 30s, after Dahlgren, who we're going to get to in just a second. Next slide. Mrs. Neal taught a field course in biology for children during those years, very famous field course, and her most, to us, famous student was, next slide, was David Rockefeller, who just died about 4 or 5 years ago. Next slide. And he was quoted saying, what's written there, I won't bother to read it to you, but basically he became interested in that at that time as a young child and stayed interested in marine biology and the environment for the rest of his life. And he lived to be 101, so he died in 2016 or 2017, right after the centennial, which is kind of interesting. Next slide. Over Dahlgren. I love this picture because Dahlgren was, one of the quotes about Dahlgren was he wore celluloid collars. Celluloid is what they made film out of in the old days, incredibly flammable. And apparently he smoked big cigars. And all of the students were sure he was going to light himself on fire during lecture because it was incredibly flammable. I don't know where I read that, but it's a great story. Next slide. He was the true founder of MDIBL, so we'll see in a couple of minutes. I'm sorry to go so fast, but I have a lot to try to cover, and I'd like to get you out of here before 6 o'clock. I hope I can make it by 5, well, it's 6 o'clock. I'll try my best. He's a true founder of MDIBL. Next slide. He was an aristocrat. His grandfather, was chief of naval ordnance, chief of U.S. Navy ordnance during the Revolution, during the Civil War, and he invented the Dahlgren gun. Next slide, please. These are 2 Dahlgren guns. Exactly. It's in Bar Harbor. It's Agamemnon Park. So we have our own Dahlgren guns. Next slide, please. He got his MS from Princeton in 1886. He never got a PhD. He was always addressed as to Dr. Dahlgren, as he well should have been, because he was a very distinguished scientist, but he was not a PhD scientist. He joined the faculty immediately after he got his MS. He had been at MDIBL while he was in graduate school. He was at the MBL, and he was the assistant director. So he worked at Woods Hole, like all good marine biologists did. And he was at the Tufts Marine Lab from 08 to 20, and director in 1920. Next slide. And he negotiated the move to MDI. Next slide. And then he became the first director of the MDI-BL in 1926. It was not called the MDI-BL until 1923, and we'll get into that in a couple of minutes. Next slide. And in 1937 to 46, he was president of MDI-BL, so he was here a long time. He must have been pretty darn old in the 50s. Next slide. And in 1938 to 41, he taught an invertebrate biology course. The MDI-BL was never really a teaching laboratory to begin with. Maybe a little bit of teaching, but individual teaching, but no coursework per se. But he had a new course in invertebrate zoology in 38 to 41. It was a big change for the laboratory. And his co-instructor was Wendell Berger, whose daughter still lives here, who must be here. He's right there. Exactly. Next slide. These are not founding parents, but they're such neat people, and I love them dearly, even though I never met them, but I just had to include them. The Lewises were at the Harpswell lab for a couple of years in the mid-teens, which we'll get to in just a second. And then in 1924, came up here to the MDI-BL. And were here fixtures at the lab until the early 1960s. Many of us, next picture, I had to show this, so I just love this picture. This is their family in the early probably 1924, 25, circa 1925. And they had 3 children. Warren Jr., but he wasn't really Jr. because his middle name was different. His middle name was Reed, which is what her first name was, or married. Thank you, dear. Maiden name. 2 daughters who many of us know or knew, they're both passed away now. Margaret, the older one, who lived to be 101, lived on a lab property for many years, on the cottage and the lab property. And the younger daughter, Jessica. Peggy, as Margaret was called, there's Margarets in every generation of that family. There are Margarets for about 5 generations, which is pretty interesting. Peggy was an astronomer at Harvard. And Jessica, who many of us knew, was a hematologist. And a very famous hematologist, wrote a textbook. Sorry for the digression, but I just love that picture so much I had to show it. Next, please. Good. Oh, yeah. Hello. Another Margaret, yeah. So Margaret got a BA at Goucher in 1901. Next slide. Thank you. From 1902 to 1907, she studied with Thomas Hunt Morgan at Bryn Mawr in Columbia. T.H. Morgan is a very, very famous biologist, got the Nobel Prize in the middle 20s for showing that the genes were on chromosomes. But in those days, he was an invertebrate development person, as a lot of people were at the MBL. So she worked with him, never got her PhD, got an honorary PhD, honorary degree, honorary doctorate about 30 years later. Next slide. She worked at the MBL in 1905, and that's where she met Warren. We'll get to Warren in a couple of minutes. Warren was an MD in the anatomy department at Johns Hopkins, but he worked for 4 or 5 summers at the MBL connection again. In 1908, she went to Germany and spent a summer there and learned how to do tissue culture. And she was the first person, as far as we know, ever to culture a mammalian cell and to maintain that cell line. So it was a huge advance. People had done invertebrates, they'd done amphibians, but they'd not done mammals. And she did guinea pig bone marrow. Next slide. 1910, she left graduate school, married Warren, and they worked together until he died in 1964. Largely using the techniques that she had initially developed. So the first 10 years or so, they basically worked worked out the specifics of tissue culture, which many people still use today. Next slide. Warren got his MD at Hopkins in 1900. He was in the 4th class of Johns Hopkins Medical School, taught by the famous 4 horsemen, or whatever they were called. And he stayed at Hopkins for many years after that. Next slide. He worked on embryonic induction. I haven't got time to describe this. If anybody wants to ask a question later, I will. And eventually had a slide explaining it. I just don't have the time to go through it. Embryonic induction was a huge deal in the early 20th century. And he was one of the pioneers. The man that he was sort of competing with, Hans Spemann in Germany, got the Nobel Prize in the mid-20s. But Warren made some incredible advances in that system. Number 2, next slide. He also did in the late 20s, picked up video microscopy, which is a very early use of that. And he discovered pinocytosis, which is cell drinking. Cell membrane extends out, takes in some of the medium, takes it into the cell, digests what's in there, and then gets rid of the excess water, if there is excess water. If I've got the time, and I hope I do, there's a wonderful movie of pinocytosis, which Warren took in the late 1920s. And as you would expect, YouTube has a copy of it. And I'd love to show it to you if I can keep talking this fast and Dawson doesn't ask any questions. Next slide. Don't be inhibited, David. That's okay. He was also very famous, Warren, because he edited 4 volumes of Gray's Anatomy, the standard anatomical text for medical schools. Next slide. Member of the National Academy of Sciences. Next slide. And they were at, as I said, they were at the Tufts Marine Lab in 16 and 17, MDI from 24 to about 64 when Warren died. He was born in 1870, so he was 94 years old, 29. I wish I had known them. Next slide. So the move to Salisbury Cove or Salisbury Cove. Now, I don't want to get too much involved in this, but I can't help but talk about this because there are some very strong feelings in this room, not looking at anybody in particular, about which one is which. Right? I'm going to try to settle it for you. I'll do my best. We'll get to George Dorr in a minute. This is much more fun than George Dorr. Next slide. There are many Salisbury headstones in the Salisbury burial site, right above Jan Merriweather's house, basically, right? And so Gene and I went up there this a couple days ago, and we took pictures of the stones just to see kind of a brief count of how many Salisburys versus Salisburys there are. Well, here are 3 Salisburys with an I. Next slide. Here are 2 Salisburys with an I. Next slide. And here are 4 Salisburys without the I. So it's 5-4, Salisbury with an I. But just to clarify things, I thought I'd show you a map that you could buy in town, which sort of settles the problem. Next slide. Note Salisbury Cove. Note Salisbury Cove. I mean, that was taken 2 weeks ago. Can you imagine that? So the point is take your pick, folks. You can do it either way. The lab's gotten away from it, I noticed on the lab website now, it says Bar Harbor. I mean, how daring is that? All right. Well, many of us have seen this quote for many years. These were remarks at the Cerdamont National Monument dedication in 1916. That's the precursor of Acadian National Park. Okay. And so George Dorr gave a speech, and in that speech, he says, he was talking about how great it would be to get scientists up here, a summer home, however simple, for men of science working the government bureau in museums and universities. And so that meant that by 1916, he was looking for scientists to move to the island. Now, here's next slide. Here's somebody none of you have ever heard of, I can bet. He was there also. He was a very famous marine biologist. He was a friend of Jessup's, a friend of other people who were in the dedication. And he was really a very famous guy. And he also made remarks, and he was more specific. He said, more desirable and practical station for such work than offered by the attractive, sheltered, and deep-watered coast at Mount Desert, now dedicated to the memory of S. Weir Mitchell. Okay, now does that name ring a bell? Next slide. He had been at Harpswell, he knew Kingsley, and he probably knew Dahlgren from the MBL. Next slide. The original plot tract was here. You know, we are now sitting right there. Original tract. The Proctor tract, which we now own, came to this laboratory in the late 1950s when Proctor died. Proctor was an early scientist working at the lab, an early benefactor working at the lab. I'll show you some buildings in a minute that he paid for, actually. And then there's the McCaig tract, which was bought in the late 20s using largely money donated or given by Mrs. Bowen, who I haven't got time to talk about, and also a couple of the scientists who were working at the lab. Next slide. So you recognize that? That's S. Weir Mitchell. You know where it is? There you go. The MDIBL has a bust someplace in some dusty corner. We all have one. Mitchell was a really famous guy. Mitchell had no connection with the MDIBL at all, but he's so interesting, I have to talk about him. Next slide. He was a Civil War surgeon. He was an author. He was a poet. He wrote novels. Next slide. He was also a pioneer and clinical neurologist. Next slide. He recruited William Osler to the University of Pennsylvania, which was the start of modern medical education. Next slide. He was the founder of the American Physiological Society. 4 scientists founded it in the 1870s, and he was the major person behind that, which many of us belong to the American Physiological Society. Next slide. And he was a friend of Morris Jessup. And he spoke at the dedication of the library. And then he died in 1914. And in my readings of Dorr and my reading of, there's a couple of biographies of Mitchell, luckily, because if there hadn't been, I would have worked on one, I think, because he's such a fascinating guy. But I saw no contact between the two men, but they must have known each other. Next slide, because he spoke at the dedication, he passed on. Next slide. And the Mitchell tract was named for him. What happened was that George Dorr and his friends, the Wild Gardens of Acadia, that group of people that started our laboratory and started Jackson Lab, basically, in terms of property. What was I going to say? It's terrible when you're in your 80s and you're in the middle of a sentence and you just lose it completely. Anyway, nah, it doesn't have it on the next slide. Anyway, so Dorr, I got it. Dorr wanted to dedicate something for Mitchell after he died. They wanted to have a plaque or something in town, at the green in town. But then they discovered this tract out here, thought it'd be a better place to name for Mitchell. So that's what they did. Next slide. So the original building was built, the scientists came in 1921 by boat. By 1922, they built a building, which looks a lot like identical, but looks a lot like the original building at Harpswell. Next slide. And it was called Old Main. It was called Old Main because the original building of MBL at Woods Hole was called Old Main. So they carried on that tradition. It's now the Neal Lab, as you all recognize. Next slide. In 1923, the Mitchell Station becomes the MDIBL. Initially, it was called the Mitchell Station of the Harpswell Marine Laboratory. And in 23, Dahlgren negotiated a name change. And so, in fact, this is our 100th anniversary of the name. Next slide. Okay, briefly, some original buildings. Next slide. Not bad. Proctor building. Proctor had the money. He was a self-made, he's not a self-made man. Can I say that? He inherited $8 million. That's not self-made. And he built some property in town. He also built some laboratories here. And his work was doing a survey of animals and plants in Frenchman Bay and also on the island. And most of the early scientists for the first 3 or 4 years at the MDIBL worked on that program. And so Proctor built some buildings. He didn't build Neal, but he built that building, which was a dark room, and then 2 structures here, which you can't really see very well, but there are actually 2 buildings there. Next picture. And here they are in modern times. This is the Neal Lab, the original Old Main. This is now Rappaport. And this is the kidney shed here, and Epstein is the one behind it, named after people who have and areas that have worked here. Next slide. Yeah, so Neal Lab, Rappaport, Epstein, and the kidney shed. Next slide. He also built the dock. Here it is in 1925. Next slide. Here it is in 2011, pretty much the same. Next slide. And I've got to show a picture of Salisbury Cove, mostly because it's a beautiful picture and because my house that I live in is right there. And it was two-tone. This is not 1919. I think that's number 19 of the pictures. It's sometime in the teens, maybe early 20s. We're not too sure. This is Dahlgren Hall, behind which we are now standing. The Baptist Church, our neighbors in our house. Yeah, our house was built in 1900. And if you look carefully inside, you can tell. Next slide. Okay, some scientific milestones. I can't cover 125 people, but I can cover some trends and try to give you a feeling for the kind of research that was done here. And also the idea that there was a progression of research. People worked next to friends and colleagues and followed up on their research a few years later. And we have a long history of doing this. I should say what I always do when I talk about the laboratory. This is the only marine lab. It's not a marine lab anymore, but it's on the shore. It was the only marine lab historically to have MDs, and PhDs working on together, maybe in the same lab, maybe not, in similar projects, communicating daily. Their kids were raised together. We had great social activities. In the 20s, they were even better social activities, I'm told. But it was according to the postmaster, who I won't quote. But anyway, the point is that no other, MBL isn't this way. Scripps is not this way. Hopkins Marine Lab is not this way. Maybe some of the European marine labs are, but we're unique in that aspect of marine scientists working alongside with clinicians.
Before you talk about these monsters, I'm going to send you back 40 years, but can you just tell people real quickly the rationale for why they wanted this last year?
Yeah, but money.
Yeah.
Does that answer your question? No, that was the main reason. The main reason for moving was that they were having real financial problems in Harpswell. They didn't have a lot of donors in Harpswell. They couldn't expand. It was right after the First World War, and Dahlgren knew people in Bar Harbor, and so he wrote to them. He knew Dover, and he knew some other people. Eno was also from Princeton, and yeah, and he also, it was a big piece of land, and so it was an obvious move, but it was mostly because he knew that there were a lot of rich people here, and so in the future he could probably use them for what was not called development then, it was called public relations. Yes, because there was no funding at that point. NIH and NSF were invented in the really partially invented, 50s and 60s. And so there was really no federal funding of any level. So everything was based upon how much a person could pay to work here, and rich lady so-and-so who would give you money to keep the lab going. We, of course, don't worry about things like that now. Oh, I'm sorry. Yeah, I was not listening. Yeah, we were the first laboratory on the East Coast to be in the boreal zone, the Arctic zone, which is cold water fish. Very different animals than you have at Woods Hole. Very different animals than you have at St. Augustine, Florida. The dividing lines are basically Cape Hatteras and Woods Hole, or Cape Cod. So we were the first northern laboratory. Sorry, I should have didn't say that. Next slide. E.K. Marshall, president of the lab from 60 to 64, probably did the most famous research, or the research this laboratory is really most famous for. He started kidney physiology here, basically. Next slide. And what he did was to show that the kidney could do more than just filter urine, filter blood to make urine, and then reabsorb salts and water. There was a controversy in the 20s whether, in fact, the kidney, besides this is a simple nephron. There are about 1 million of these in each of your kidneys. They're microscopic, which has a filtering system called a glomerulus, and then a tubule here, a tubule there. That's only in mammals, little ones in birds, and distal tubule in the collecting duct. A fish basically has this. For those of you who care about what fish kidneys look like. So the controversy was, can the kidney secrete salts, or can it secrete solutes into the urine as well as reabsorb it? Okay. And so the next slide. So I already said that. Next slide. So Marshall did some reading. What he wanted to find was an animal that didn't have a glomerulus, that did not have a filtration site. If you just had a tubule without the filtration site, then in theory, things could be added to the urine. So we knew that there's aglomerular kidneys without a glomerulus in certain species. Goosefish was a good example. Next slide. And he had a neighbor in Baltimore, Warren Lewis. And they were having dinner one night, and Lewis said, "Oh, you ought to come to the lab in Maine because I eat the tails of monkfish," which is another name for these animals. "I eat the tails all the time," which reminds me, the new restaurant in town, the French restaurant, has monkfish on the menu. which I'm told by some people is not very good. It's only a sample size of one, and it was just Dawson. So, how much taste does he have? All right, so next slide, it should be a picture of this lovely animal. I mean, who would want to kill an animal as cute as that? And the first couple of years I was here, they were still catching them. They don't catch them around here anymore. I'll bet they're not catching them locally for the restaurant. They're a deep sea angler. They're just wonderful animals. So that next slide. So what he had now was an animal without a glomerulus. They're aglomerular. Next slide. Oh, never mind. What he found was he measured the urine flow and the composition. It was comparable to what you find in glomerular species. But what was really neat, when he injected phenol red, which couldn't be filtered now, he injected phenol red, and phenol red appeared in the urine. So it had to be secreted. And so this was the first definitive proof of renal secretion, which we all know, we all spend days in our lives now thinking about it, of course, but it's a very important phenomenon. So the kidney filters, reabsorbs, and secretes, all three functions. Well, when I get done, can you sing it for us? Well, I'd say there, but I gotta get through all these first. Next slide. Brings us to Roy Forster, one of the true gods of the laboratory, and one of the true really nice people. I was lucky enough to know him. Many of us in this room knew him also. He came to the MDI-BL by pure chance in 1937 because he went to the Federation meetings. He had worked on uric acid secretion in birds. And he went to the Federation meetings, and Jim Shannon, who was a very famous renal physiologist in those days, who had been at the laboratory, who became the head of the modern NIH. The modern NIH main administrative building is called the Shannon Building. So Jim Shannon, who I never met, gave a talk and basically scooped everything he talked about. Poor Roy had in his dissertation. Everybody's worst nightmare. He was entirely scooped. And Shannon felt so badly about that he invited Roy Forster up, young graduate student that just finished his PhD, come to the lab and meet and work here. And Roy came in 1940. And he was present in 70. He probably stopped coming in the middle 80s. And then since he's passed away, obviously. So he came to MDI 37 because of Shannon. Next slide. And during the World War II, he was a director, and he basically kept the laboratory alive, kept it open. Got the dock replaced in 1947 from the Thorndikes. And just really, if he had not been there, the laboratory would have probably gone under. And then he was president in 64 to 70. Speaking about when I came to the lab, I first came in 65 when I was at Woods Hole to meet Roy Forster and Leon Goldstein. And I think I met Wendell Berger too. I'm not positive. I must have. Came back in 66 when Roy Forster was retiring, came back for his symposium. And then I came back in 76, sorry. And then 78, I came back and have never left, except in the winter when I go home. Next slide. So his seminal discovery was the isolated fish renal tubule. This is a huge step forward. Until Roy did this, nephrologists could study what was going in by injecting it into the blood. They could measure what was coming out by taking urine samples, but it was a big black box in there. So what Roy did, next slide, was to isolate renal tubules and then put them in various kinds of solutions and see if they would reabsorb what it was in the solution. Next slide. There was no glomerulus. Very famous picture. Here's an isolated fish proximal tubule. Phenol red is out here and phenol red is inside, showing directly there was secretion, but also it's a tractable system that one could use to measure things going in and out, electrical potentials across the membranes. I mean, it was a huge step forward. And people are still using that procedure today. Gert Fricker's in the back. He still does it every summer. Next slide. So this was the basis for perfused tubules. Next slide, which allowed one to put pipettes on either side. You could control flow through. No, next slide. You could do measure various electrical parameters. We'll have Dave Dawson explain the slide when I get done with it. Because only he enjoys things like that, not the rest of the stuff. But anyway, you can measure fluxes of ions, you can measure electrical potentials, you can measure currents, you can do all sorts of neat things. And basically, next slide, this was the basis for the next 50 years of research in renal tubular transport. That simple teasing out from the fish kidney tubule, from the fish kidney tubule. Next slide. Homer Smith. Next slide. Homer Smith was Marshall's technician. Marshall was in the army and during the First World War, working on mustard gases for obvious reasons. And he had a technician he hired who was an undergraduate, and it was Homer Smith. It was Sergeant, it was sort of Captain Marshall and Sergeant Smith in Washington, D.C. Next slide. And Marshall helped Smith go to graduate school at Hopkins, and he got his Doctor of Science in 1921. He published 13 papers by then. I mean, I was pleased to have published 2 or 3 papers for my dissertation. He had 13, which is kind of amazing. Next slide. He worked at the MBL, like everybody does, worked at the MBL in the 1920s. And the funny thing is he worked on invertebrate fertilization and development, did not work on the kidney. Next slide. Came to MDI-BL in the mid-1920s before E.K. Marshall came. Homer Smith was here first. Next slide. And by 1927, he was a fixture at the laboratory and was a fixture until the 60s when he passed away. Next slide. He studied, this will take some doing now. He studied both bony fishes and sharks. Next slide. And he worked on osmoregulation, salt and water balance. And I'm going to go through this pretty fast, but it should be fairly straightforward to you. Next slide. The shark was the logo of MDI-BL until about 10 years ago. Show you how important it was to the laboratory. That's like one of the original signs. It's mine. I have it on my back porch, and that was the pretty one. I liked the pretty one. Next slide. Shark blood has less sodium chloride in it than seawater does. That's true of all vertebrates in the marine environment, except for hagfish. But they're an odd duck anyway. But an interesting one. So Smith knew this, and so the next slide. Smith proposed that the only way the animal was always going to be facing a salt influx, because it had to breathe through its gills, had to be permeable and highly perfused. So it was gaining salt by diffusion, okay, because that's the gradient, and it must excrete it somehow. And Smith proposed, quite rightly, or thought quite rightly, that, well, vertebrate kidneys can make saltier, and we can. So maybe all vertebrate kidneys can do that. Next slide, Roy. The trouble is they can't. The urine sodium chloride concentration of sharks and basically any marine fish is about equivalent at best, about equivalent to what the salt concentration in the body fluids are. Next slide. So Smith proposed there must be some extrarenal excretory pathway. Next slide. And that brings us to Wendell Berger, who was the teaching colleague of Dahlgren in the early 1940s. And he usually worked on birds, on bird reproduction. But by the 1960s, late 1950s, he started working on sharks. I stopped myself from telling you other stories, but started working on sharks, and he cannulated the dogfish rectal gland. Now, the rectal gland, next slide, is a gland that empties into the intestine. It's not a kidney. It's a digitiform gland about that big, and a shark about that big. Close enough, Patricia? And this gland, and the anatomy of this gland had been known for a long time, but nobody knew what it did. Wendell Berger cannulated it and found out that, lo and behold, this was the extrarenal non-kidney pathway. It secreted salt, more salt per volume, even than you find in seawater, which is really extraordinary. Next slide. And this began 60 years of research at the lab on the shark rectal gland. If there was any, it was probably the more people worked on rectal gland than any other single tissue. I think that's probably right. Next slide. And here's the 4 horsemen of the rectal gland. Frank Epstein, Wendell Berger, first of all. Frank Epstein was even then a very famous nephrologist from first at Yale, then at Harvard. And he brought up in the early 1970s 2 very young-looking fellows, Patricio Silva, who's here today, and John Forrest, who unfortunately is not. And one could spend a day talking about their research. One is not going to do that. All I can say is that they worked out the basic physiology and the neurohormonal control of the gland. All the proteins that were involved, all the stimulatory neurons as well as stimulatory hormones. They also had, next slide, they also had 3 colleagues who worked with them, Jeff Stoff, Mike Field, and Rick Solomon. Stoff and Solomon were also fellows of Frank Epstein at Harvard. Mike Field was a gastroenterologist at Columbia, and he did a lot of talking to these guys and came up with the models that we all agreed upon. Next slide. And in addition to that, by the late 1980s, people were doing things like tissue culture and perfused tubules. Reiner Greger came up here and used perfused tubules and confirmed most of what these guys had already found out with regard to transport proteins. John Valentius came up only for a couple of summers and tissue cultured the rectal gland, which is a big deal. He unfortunately only came a couple of summers. Next slide. Next milestone, thiazide diuretics. Some of you in this room, I know at least one is, take thiazide diuretics. Larry Renfro, biologist, just like I was. He worked on flounder urinary. He was a postdoc of Bodil Schmidt-Nielsen's and then stayed on for a few years. So in the 70s, late 70s, he worked in the flounder urinary bladder, which I won't spend a lot of time on, but to make a long story short, it mimics the human distal tubule in the kidney. It does the same sorts of transport things. Just take that at face value. Next slide. And what he showed using radioactive isotopes was that the fluxes, the movement of sodium and chloride were very similar. So similar, he said they must be a co-transport system, which in those days was pretty much unheard of. It was just being discovered of co-transports as a class of proteins. Next slide. So this is one of the first descriptions of coupled transport. Next slide. There he is, folks. Dave Dawson. No, it's Dave. That's a story we won't get into. Dave Dawson can't get him to smile very frequently. He did all of his obscure electrophysiological measurements, and they basically came up with the same conclusion, that sodium and chloride were coupled. The next summer, Dawson was gone, and Dawson was nice enough to loan a new investigator his equipment. Next slide. New investigator was John Stokes, who was an MD. Dawson's an electrophysiologist. John Stokes is an MD. He came up for 2 summers, I think. And what he showed was that this coupled transport in the flounder urinary bladder was inhibitable by thiazide diuretics. So the thiazide diuretic, which had been in use medically, they didn't know the site of action, didn't know what protein it was inhibiting. Turns out it was inhibiting this co-transport system. Classic case of the progression of research at the lab using a marine species to look at a fundamental process. Next slide. So next step was molecular biology. Next slide. Ed Benz, who unfortunately is not here, but I think he's online. He brought in 1988, and I never forget when he came. He came he was our molecular biologist. We finally found a molecular biologist, which was just emerging in those days. And he gave a series of lectures that could have been given to undergraduates, because most of us really weren't trained in molecular biology at all. You know, what's messenger RNA kind of, we're at that level basically. So he gave a series of lectures, also did some molecular biological research here. Next slide. In the early 1990s, Steve Hebert from Yale came and basically sacrificed, sacrifice is a nice word for kills. They sacrificed as many flounders as he could and got their bladders and took them back to Yale. And he cloned for the first time the thiazide sensitive cotransporter that we've been talking about. One of the earliest, I think, proteins, that's probably right, one of the earliest proteins, at least from the kidney, that have been cloned. Next slide. Biff Forbush was about the same generation, although Biff was here many more years than Steve Hebert, and he cloned another transporter, another co-transporter. In this case, it's potassium and sodium and 2 chlorides are carried, not just a sodium and a chloride. And he cloned that from the shark rectal gland. And that's the protein that's inhibited by loop diuretics, which some of you may be taking, Lasix and furosemide. Not bad. I'm not done yet. Next slide. Well, I got to show you where these things act. Here's a much more modern, much more detailed human kidney: proximal tubule, loop of Henle, distal tubule, collecting duct. Next slide. The site of action of loop diuretics, furosemide, is this cotransport system in the thick ascending limb. It's not exactly the same as the one that Hebert cloned. They're from the same gene family, however. Next slide. And the thiazide diuretic acts here, inhibits sodium chloride. They both inhibit sodium chloride resorption from the kidney. Therefore, your urine has more sodium chloride. It lowers your blood pressure because you reduce it. You keep the blood concentration of salt low. Next slide, please. Well, I haven't mentioned a woman yet. Now, science in the late 20th century didn't have that many women. I mean, comparatively speaking, things have changed, thank goodness. Didn't have that many. And we had 4, 5, 6 come up, and that was about all. 3 of them were here for many years and did really important work. The other ones did important work too, but let me just mention 3. Next slide. You got to talk about Bodil Schmidt-Nielsen. She was a very famous scientist. Her father was a Nobel laureate. She was Danish. She was recruited by Homer Smith in 1952. Next slide. She worked on amphibian kidney function, especially urea transport. Next slide. And she liked it up here so much that after she got divorced, she moved here with her second husband, moved here in 1970. So she and Bill Kinder, who came the year later, 71, they were the first year-round people at the laboratory, and the last until we started our new generation after the turn of the century. Next slide. She was the president of MDI-BL for 4 years. Next slide. Is also president of the American Physiological Society. She was the first woman. Next slide. The next woman president was in 2007. That's the bad news. Next slide. Here's the good news. 7 out of the last 10 women presidents of the APS have been women. So the times are changing. Next slide. And there's also a Distinguished Mentor and Scientist Award, which the APS gives every year. So she's a very beloved by all of us who know her. Next slide. Ruth Sayre, or Helen Sayre, Ruth's daughter, sorry. Helen Sayre was recruited by David Roll in '66, came and worked, next slide, on cerebral spinal fluid formation, the dogfish and the skate and the hagfish. Next slide. And she also worked on the blood-brain barrier. Next slide. And Gloria Callard came, she and her husband came, they're both from BU, she came in 1979. Next slide. She studied endocrine control of reproduction in dogfish and eels. They were here for probably 10, 12 years, I think. Next slide. She worked primarily on steroid hormones in males. Next slide. Which I thought was interesting because her husband worked on steroid hormones in females, which I thought was kind of backward, but I always thought that was interesting. Next slide. Okay, we are getting toward the end. This is good. Next slide. We've had a lot of really good administrators here, and there are 2 that I want to bring to your attention. One is Patricia Hand. Next slide. We recruited Patricia in 2000. We were incredibly lucky to get her. She was administrative director. We'd had 3 administrative directors before her. She was very well qualified. Next slide. She'd been at the NIH for 20 years, knew the NIH in and out. What a huge advance for us. Next slide. So she coordinated submission of the first big grant, which is called the Brin grant to the NIH. And this was a grant, next slide, which would coordinate research and teaching between many individual universities and colleges in Maine. And it was awarded in 2002 and it was $5.5 million, the biggest grant this laboratory had ever gotten by that point, by a long shot. Next slide. The Brin grant became what's now called an Embry grant. Next slide. And the total that grant has generated is $87 million for this laboratory. And that's one of the primary things that keeps this lab going. Next slide. Ah, there she is. I'm gonna tell you that I asked Jerry 3 times for more information. And I'm gonna give you is all she's given me. Because she just didn't have the time to tell me much. That's all right. So she came from the Jax Lab in '99 as development officer. We had a whole series of development officers, small d, small o. She was our first and now ongoing development officer, capital D, capital O. Next slide. She brought, what was really neat is she brought an interest in state and a knowledge of state officials and connections to state officials, both in terms of state legislatures and the governor, as well as state officials who were senators and congressmen. Next slide. So she built a really professional and active development office. And we now have 4 or 5 people in that office. Yeah, well, that's good. And you have one leaving. Next slide. So here's all I got for you, folks. Is the endowment was $1 million when she came about, and now next slide, it's about $5 million, and that's as much as you gave me. Yeah. But I think it's extraordinary. Next slide. I think I'm almost done. Yeah. Made it. I told you I could do 150 slides, Dawson. You didn't believe me, did you? There's where we came from. There's where we are. Next, no, leave that. This is the end. Here, can you stop that, Roy? If you've got time, I'll show you a video. This is. We probably have the time. This is Warren Lewis's video that's on YouTube of pinocytosis, of cell drinking. It was taken in the late 1920s. The only downside of it is it takes about 2 or 3 minutes to get through all the jiggly things to get to the cells. Now, do you want to take the time to see it? Sure. Okay. Okay. Yeah, and then we can have the song after that. Yeah. Yeah, well, only 5 minutes away. So this will take a few minutes. So I can answer a couple of questions while you have any while this loads. Anybody have any questions? Well, that's good. Then moving on, we'll do something else. I mean, it's wonderful. You got to remember, this is like watching Birth of a Nation, basically. This is not a talkie. And anybody can get this on YouTube. There's a couple other ones of his movies on YouTube. Maybe later, over a beer. They must have held these things up, and the camera was jiggling. It's really cool. He's got another one of locomotion and macrophages, which is pretty neat. But these movies were shown all over the world. They're very, very famous movies. Because they were the phenomenon is famous, but also just the fact that you could take pictures of living cells at 32 times the speed. So here's the cell. It's not as obvious as you may think. You see these extensions going out? Those are called pseudopodia, pseudopods. And they basically are engulfing the medium and then bringing it in, and it forms these vacuoles, which here. This is the nucleus. So it's forming these vacuoles, which are then, if there are proteins in it, they can be reabsorbed. If it's just fluid they need, they can absorb that. I love the analog non-digital timer. I have no idea. I don't know. Okay, that's probably enough. I mean, how much fun is that? Roy, you can stop that now if you want to. Okay. All right, I'm not going to talk about little unless somebody asks me.
What about little?
No. All right, 2 minutes. All right. Little's the father of the Jackson lab. Okay. The Little came through our laboratory. We are the home of the father of the Jackson lab. that fair enough? Next slide. So in the middle 1920s, he was president of the University of Maine at Orono. And he got some land from George Dorr just south of Bar Harbor. It's right next to where the Jax Lab is now. I'm sure the Jax Lab is probably grown over it. They got some land because he wanted to have a summer field course. Next slide. And that facility was called the Dorr Station of the MDI-BL from 1931 to 1941. It was part of our laboratory, summer teaching. Next slide. And this is what you probably don't know about him. Is in 1928, he was served as on the Board of Trustees from 28 to 36. And he was our president from 31 to 33. It's so far buried in our archives that his picture's not up there, and it ought to be. I mean, he was a very controversial man. Those of us who know his history, he's very controversial in his later days. But the reality is he was one of our presidents. Next slide. The important thing he did is he actually saved this laboratory. In the late 1920s, Rockefeller was on our board. Rockefeller was on the board at MBL, at Woods Hole. He was convinced that the two institutions should merge. Really, we would have been subsumed by the MBL. And Mrs. Bowen was on board with that, apparently. But the good thing for us is Little was not. And so Little talked against it so much, and it's such a good arguments against it, that the lab never voted on whether to do it or not. So the lab didn't do it. But if we had, we would not be here, I don't think. That may be the last slide. Any more? Anyway, so in the 1930s, he then came back as the first president of the Jax Lab. That's it, Roy? Yeah. Now, if we can all just go have a drink and then Judy can sing for us. Seems fair. You want to sing the song now, Judy? That's a short song.
Olopius piscatorius. What happened to your glomerulus? We searched the bottom of the sea, but we could find no fish could need. Olopius piscatorius. You still have no glomerulus.
Thank you, dear. All right, thank you all for coming. I appreciate it.
Thank you, David.
You're welcome.
Thank you. Just a quick note, for those of you that may be interested, we do have an entire week of activities planned around the celebration of our 125th anniversary. And David, I just want to take a moment to acknowledge the tremendous amount of work that David has done prior to this anniversary and developing that amazing history of the lab. But most, it's all about me, most recently just been an amazing resource to my team as we thought about, the significance of this anniversary and how we might want to acknowledge it. So I really, really appreciate all the effort that you've put into that. And for those of you that are interested, as David said, he had an incredibly difficult job to try and truncate 125 years in just an hour's presentation. But we have put together a nice timeline in this issue of Breaking Through. And then there's a small takeaway one here that kind of folds out. And then next on Thursday, we'll be debuting our new online timeline. So a really nice sort of interactive timeline on the website that encapsulates some of the 125 vignettes that David wrote about each of the individuals who've been here. We'll add to that over time, but we were kind of pressed to get it out there for the annual meeting. But in any case, lots of fun things. So I encourage you all to come for the annual meeting, which is on Thursday. It starts at 10 a.m. We'll have a nice little look back at some of the accomplishments but really talk about where the lab is headed in the future, because that is kind of the fun and exciting part. I can't wait. I often think about what some of those early scientists would think about some of the technology that we have today. So we're excited to share that with you, and we hope you'll join us. Thank you all for being here tonight.